Hello, thank you for joining today's Biodesics-sponsored webinar on the utility of a new targeted next-generation sequencing test for liquid biopsy samples from patients with non-small cell lung cancer. My name is Natalie Todd, and I will be your moderator for today's presentation. Before I introduce our speaker, I have a few housekeeping items to mention. First, this webinar is being recorded, and a link to the recording will automatically be sent to all registrants and attendees. As attendees, you are in listen-only mode. If you have a question you would like to ask, please type it in the chat box, which should be accessible when you select the chat icon at the bottom of your screen that appears after you move your cursor towards the bottom middle. I will collate questions at the end of the presentation and read them out loud to our speaker. Without further ado, it is my privilege to introduce today's speaker, Dr. Hestia Mellert. Dr. Mellert is a senior director within the Biodesics Development Group. She has a background in molecular cancer biology and experience leading teams in the development and commercialization of blood-based and now respiratory-based diagnostic tests under CAP, CLIA, and New York State CLEP. Dr. Mellert has played a leading role in the launch and on-market support of the GeneStrat tests, which uses ddPCR technology. Most recently, she has contributed to the expanded use of this platform at Biodesics to support molecular testing for SARS-CoV-2 infection. She continues to focus her efforts on bringing relevant and actionable genomic and proteomic information into the clinical laboratory to support pharmaceutical development programs and physicians. Dr. Mellert received her PhD from the University of Pennsylvania and postdoctoral training at the University of Colorado. She has received several honors, including the American Cancer Society Postdoctoral Fellowship and inaugural Biodesics Invent Our Future Award. With that, I'd like to pass the mic over to Dr. Mellert for her presentation. Thank you.
Thank you so much, Natalie, for that really kind introduction, and for all of you that have joined today to share with you our newer technology for genomics, next generation sequencing. We'll start out by giving a brief introduction to Biodesics, and then move into the background on the GeneStrat 52 test, which is a test that uses next generation sequencing to look more broadly from liquid biopsy specimens at the genome. Then we'll look at feasibility and development efforts that went into initially bringing this test into the clinical laboratory. And the test can measure SNVs, indels, as well as fusions and copy number variation. Following that, we will talk about the validation of NSCLC driver mutations using this assay. And there, the focus is going to be on DNA variants, including SNVs and indels in EGFR, KRAS, and BRAF. And then finally, to close out the talk portion, we'll go into the clinical use cases for EGFR monitoring. Biodesics is a lung cancer diagnostic solutions company. There are two core business units. The first I'll talk about here is in the white circle. This is our on-market clinical testing focused area. And there, as Natalie spoke to a little bit, we have multiple on-market tests that are aiding physicians in decision-making in lung disease and cancer. The first two here, Notify CDT and Notify XL2, inform about indeterminate lung nodules. The second, GeneStrat and VeriStrat, are combined to facilitate a reflex strategy that is used following diagnosis of lung cancer. GeneStrat is specifically looking in liquid biopsy specimens for actionable mutations that are associated with NSCLC. And VeriStrat provides additional information from the serum proteome that allows you to further prognosticate. In addition, we have two pretty new tests here just in the last few months as the SARS-CoV-2 pandemic has come. We realized that we have very good technologies already established in our clinical laboratories to approach these needs of being able to identify both infection through molecular testing. And so we have launched our Bio-Rad SARS-CoV-2 ddPCR test into the Biodesix Clinical Laboratory. And then also we have high throughput ELISA capabilities, which set us up to be able to quickly implement the Platelia SARS-CoV-2 antibody or serology test. So in addition to the on-market clinical testing side of the business, we also support pharma through partnerships where we use these same technologies. On the pharma side of things, we don't necessarily need to focus just on lung or just on cancer, and we support many different biomarkers and different indications, all the way from preclinical research through clinical trials. This slide gets a little bit more into those technologies I was just speaking to. One of the unique things about Biodesix is our ability to join genomics and proteomics. And you really use different types of technologies complementarily to each other. So the first is droplet digital PCR. This is a technology from Bio-Rad. They have FDA clearance of their platform for BCR-ABL testing, and also an authorized test for SARS-CoV-2, as I just mentioned. This platform is really wonderful for looking at targeted mutations or expression tests when you really know already the biomarker of interest. In addition to that, we have our NGS technology, which is the Thermo Fisher Ion Gene Studio S5 systems. And here we really find that the NGS complements our target approach by droplet digital PCR because it allows us to look a little bit more broadly. And then on the proteomic side, we can complement looking at the genome by looking at the proteome using MALDI-TOF mass spec, LC-MS, and ELISA-based assays. This is a more detailed view of the different types of assays that we are capable of running in our Boulder and DeSoto laboratories. You can see the bold are those that are currently on market tests that are being offered to physicians and also being used with pharma as well. And many of them, you can see by the asterisks, also have New York State CLEP approval. So we do have two laboratories, the corporate headquarters laboratories in Boulder, Colorado, and that laboratory has CAP accreditation and CLIA certification. It's New York State CLEP certified for both soluble tumor markers and molecular cellular tumor markers. It also follows ISO 13485 with a certification. And this really does make it best in class in terms of clinical laboratories in the US. And if you actually look across the US at the different clinical laboratories, really only about 1% of these laboratories have these different types of regulations that they are following. And then the second is in DeSoto, Kansas. This is a newer laboratory for Biodesix. And that is where the nodule tests are run out of, and also some additional pharma work. And there's a COLA and CLIA certification, as well as New York State CLEP certification. So now that we've introduced Biodesix as a company, I wanted to start getting into the topic for today, which is the GeneStrat 52 test. So as I mentioned, this is a next generation sequencing based test that runs off of the Gene Studio S5 platform from Thermo Fisher. We have the Prime platform specifically in our laboratory. And here we're talking about the Oncomine pan-cancer cell-free assay, which is a research use assay that is available through Thermo Fisher. The assay is designed specifically for use with liquid biopsy. It needs 20 nanograms of input material. It's a single library prep that can look for both DNA and RNA. There are molecular barcodes, which are used to allow for molecular level analysis of the data. You can batch up to 15 specimens with a single control, and the panel covers 52 genes. So within this panel, as you can see in the table below, we can measure both hotspot mutations. We can look over the exons of the common tumor suppressor genes for mutations in those genes. We can look at copy number variation in some of the common genes, including HER2, MET, for example, which are really important in non-small cell lung cancer. And then we can look at gene fusions as well. And there's actually 96 different fusions that can be detected by the assay. Here we're looking at the major fusion partner in the table below. This is what the workflow looks like as a sample moves through processing at Biodesix. It starts with shipping. We have focused this on specimens that have been shipped at ambient temperature using the Streck cell-free DNA tube. However, the assay is definitely also compatible with plasma samples that are collected in other ways. We ship those specimens at ambient temperature overnight to the Biodesix laboratory, and we accession them using our secure LIMS system. And then we can extract total nucleic acid from the specimens using standard QIAGEN circulating nucleic acid kits. Then we prepare our library. This assay currently is a manual library prep, and again, can use molecular barcoding. Once the library is completed, then the samples are moved into a templating step, which is unique to Thermo Fisher's technology. And that happens here on this Ion Chef instrument. And then from there, it moves into sequencing and bioinformatics analysis. At that point, the bioinformatics pipeline is able to identify the different variants of interest, as well as the allele frequencies, which we can report out either in batched format or as individual reports. The next slides are going to go into the validation, feasibility, and then validation of the assay. So first, let's talk about what we did to prepare ourselves prior to trying to validate the assay. Of course, feasibility is the first step. So we initially demonstrated that the assay can be implemented at Biodesix with the instruments that we had in place. We have 2 workflows currently in the Biodesix laboratory. So we were able to do inter-run, inter-instrument precision and begin to look at the accuracy of the assay as well, looking at the orthogonal methods that we have in the laboratory, that being ddPCR. And then we evaluated the cutoffs and started to really hone in on the assay's performance as we needed to set it up for validation. So once we've done that, or at the same time, really, we're also optimizing the workflow to make sure that the end-to-end process that will be locked for validation is essentially at the best point it can be, both from a process perspective and from a performance perspective. And so some of the things I've listed here that we've done is implementing a 96-well format, and we've demonstrated we can do that without crossover contamination. We've identified the specifics of the bead cleanup and drying times that are required in the Boulder area in a dry climate. And we've also identified specific areas where we can improve things like quantifying the library using qPCR to really increase the performance and the balance of the libraries as we're sequencing. Once the test is optimized, of course, then we need to lock down the process that we're going to use for validation. So this is in SOP drafting, reviews and approvals by our laboratory directors. And this SOP suite then allows us to perform everything from receiving a specimen in the laboratory to being able to report out a result. We also needed to establish the quality controls that we were going to use, and we looked at both batch level controls as well as the specimen level thresholds that would be implemented to pass specimens as they are being analyzed. And then finally, We needed technical competence of the staff in the laboratory. And so we had 2 additional operators, as you'll see later on, be trained to the process and observed by the lead scientist. And then they do multiple process verification runs from end to end using the locked SOPs to demonstrate the assay's workflow is robust before we enter into validation. So these are all things that that you may already be familiar with if you're in the field of developing and validating assays, but really important things to do before you move into validation. So let's go into the details a little bit of what we did to meet these different goals. This first slide discusses the technical feasibility that was performed on the assay. This was using an analytic control that you can buy commercially from Seracare. Actually, 2 of them that we mixed together, I should say. So the first one is the ctDNA complete mutation mix, and that one contains both variants, SNVs, and then we mix that with the RNA mix, which has fusions of interest. So really nicely, using these types of analytic controls allow you to look at many of the different types of mutations and variants that you might be wanting to understand the performance of early on in the experiments. And so you can see all of the different variants that are in these controls. And then you can see here what was expected, as well as what we were able to detect. And so we could detect all of the fusion variants as well as MET exon 14 skipping using our locked assay. We were able to detect the 2 copy number variants or gains that were in the control. So that was HER2 and MET. And then we were also able to detect the various nucleotide changes and amino acid changes that were in the assay as well, with one exception here where we missed KRAS G12D. And this is an interesting thing because actually G12C and G12D are at the exact same location in the genome. And so we have identified that this actually has more to do with bioinformatics of having an analytic control with all of the different variants in one mixture, which is not very common in a clinical specimen, rather than a performance issue with the processing of the specimens themselves. So this is a graph then that represents 2 different runs of that analytic control that I showed you on the previous page, looking at the allele frequencies for the SNVs and the indels. And what you can see here is between run and feasibility, we saw a really nice correlation and reproducibility of the instrumentation, which was a really promising first step for us. And even when you look at the low allele frequencies, which are really the critical places to be focused when you're developing a cell-free assay, you can see even there, inter-run performance of the assay was quite good. In addition, here I've graphed the fusions. So you can see 2 different runs and looking at the copies of fusion that were detected on run 1 and run 2. So these are total copies of the fusion variant that were detected and included the fusions that are in the table. And then finally, what we did was we wanted to take a quick glance at how our NGS cell-free assay compared to the ddPCR assay called Genistrad that we already have New York State approved and also in the clinical laboratory. And what you can see here again is when you're comparing the allele frequencies of these remnant specimens, so these are actually clinical samples from NSCLC patients. When you look at their allele frequency of their variants by ddPCR versus by NGS, there's really nice correlation in the results. And here you can see that same data in a table format. You can look at the different variants that we were looking for in these different clinical samples, as well as probably of note is the various levels of input that you actually can use with the assay. So I mentioned on one of the previous slides, you might have noted 20 nanograms. And this is an important input amount if you want to consistently be able to see variants at sub 1%. However, the assay can actually take less or even more, up to 50 nanograms of input, and that can have an impact on sensitivity, but can still give you very useful information. For example, here, even at 3.7 nanograms of input, we were able to detect not only EGFR del19, but also C797S and T790M. And C797S here, for example, was less than 3% allele frequency. Additionally, in feasibility and development, we really needed to understand where we were going to place the cutoff for our clinical test. And so we did some work to look at replicates of variants from clinical specimens at the input concentration of 20 nanograms of total cfDNA. And then we looked for the allele frequency that allowed us to hit 95% or greater. And so at 0.4% allele frequency, when we test EGFR del19, L858R, T790M, BRAF B600E, and KRAS G12C containing specimens, you can see we hit 72 out of 80, which was 90%. Once you get to 0.5% allele frequency, 0.6 and beyond, you start to see that your hit rate increases to above 95%. And so this type of experiment allowed us really to kind of focus in and determine what the allele frequency cutoff should be for our assay. We've determined to select 0.5%, as you'll see as we move into our validation. In addition, we needed to set control metrics for the assay. So here you can see both batch level controls were selected. We also defined the thresholds that we would need for specimen QC. At the batch level, we run one control with each batch of 15 clinical specimens for a total of 16 specimens per run, which is on two 550 chips. The positive control is a combination of admixed specimens that contain L858R, T790M, exon 19 deletions, KRAS and BRAF mutations. And there has been established cutoff now at 0.5% that all of these variants need to be called above that threshold for the batch to pass. And then on the specimen level, there's QC pre-analytically, as I mentioned, to have 20 nanograms of input. And then additionally, the sequencing metrics here, total map reads need to be greater than 100 million. The coverage uniformity must be greater than 95%. Molecular base uniformity greater than or equal to 80%. Our mean read length needs to be equal to or greater than 80 base pairs, and our library concentration needs to be greater than or equal to 50 picomole. Okay, so with that, we were in a good place to begin the validation studies. And so I'm going to give you a brief description of the results for each of those. First, analytic sensitivity and specificity, then within lab precision, and then we'll go into accuracy. Analytic analytic sensitivity and specificity. So the experimental design, we used 5 remnant clinical specimens that were each positive for a different variant, close to the limit of detection. And the variants you can see are listed there. The clinical sample that contained T790M, as it's a resistance mutation, also carried L858R. So you can see there's a study design over multiple days, so 4 days, 2 different reagent lots, flipping between operators, between instruments, and then running 3 replicates per clinical specimen. Here are the results of that study. The variant level performance using our predefined 0.5% allele frequency cutoff was 100% for analytic specificity and 100% for analytic sensitivity, which essentially means that every single time we got the right answer in this study. One note is that we had one sample that was excluded from the study because it did not meet the QC criteria that I had shared with you earlier. And then we also can say from this study that the variability between the different lots, operators, and instruments was very minimal. And you can see actually the average and the standard error of the mean for each variant here on this graph on the right-hand side. Next, we performed a within lab precision study. Here, the design was to mix specimens so that we could look at both 3x the LOD and 1x the LOD for each of the variants that we were validating. This study was done over 10 days with 2 operators, 2 instruments, 6 clinical specimens, and 2 replicates per. And here you can see a comparison of the operators as well as the instruments in the graphs below. So at both the low and the high concentration, so 1x and 3x for each variant, the allele frequency is graphed, and then the standard deviation between the 2 operators or within the operators is graphed. And you can see that they're very similar, and there was no statistically significant difference between either operator. And then the same is true for the inter-instrument analysis. When comparing the 2 different instruments to each other, you can see they're very, very similar in their performance. And there was, again, no difference between them. Finally, accuracy. We ran 51 remnant clinical specimens that were retrospectively analyzed by NGS. These specimens had a droplet digital PCR result associated with them. So that included looking at 121 variants of interest in the specimens. And should note, it's very important that these specimens are blinded to the staff that was running the study prior to them processing the specimens. And so here is our accuracy results as we compare to the GeneStrat ddPCR clinical test. You can see our negative predictive agreement between the 2 tests was 100%, so they're both very specific. And our positive predictive agreement was 91.7% relative to ddPCR. And you can see here also, if we graph the allele frequencies between the NGS and the reference result that we had for ddPCR, very similar correlation between the 2 assays. So I will briefly then, in the next couple of minutes, go through a clinical case report that was originally focused using the droplet digital PCR platform, but we retrospectively went back and analyzed these same samples with the next generation sequencing assay to demonstrate the use of either of these types of techniques. So this was a female never smoker that presented with new onset seizures. And she was diagnosed with brain and body metastatic positive lung cancer, lung adenocarcinoma, and she had EGFR del19 mutation. So she was initially treated with whole brain irradiation and also with the EGFR TKI or lotinib. And she had an excellent response initially, but as many of us know, that EGFR T790M mutation emerges upon treatment with these first generation TKIs. And so she was then switched over to osimertinib. Of course, osimertinib as well can cause resistance mutations. And so she then again had an emergent mutation, this time EGFR C797S. At that point, the decision was made to discontinue therapy and her disease was progressing. So we wanted to show you some longitudinal monitoring using both our ddPCR and our NGS approach of this patient. So at time 0 is when she initiated osimertinib treatment. And you can see here that she had already acquired her T790M mutation, as well as having the del19 mutation. And she had a response, a full response at the molecular level. The EGFR in her liquid biopsy specimens went all the way back down to below detectable. However, at about just over 150 days, we started to see a recurrence of both the del19 EGFR mutation, the C790M, and then we started to also see C797S coming up here. And you can see her scans as well demonstrating the recurrence of her disease. Now, since this talk was focused on next generation sequencing in our GeneStrat 52, I wanted to show this slide that demonstrates in these same specimens and overlays the results. So these are the allele frequencies at those later time points. I'm going to flip back to the previous slide. So we're looking at these time points here, now overlaying the NGS and the ddPCR results. And it's actually hard to see. The ddPCR results are in blue and the NGS results are in orange. And in some cases, you can't even see the ddPCR results because there is such great concordance between the two assays. They're both very good at looking at the monitoring and increase in EGFR del 19, so demonstrating the ability of the NGS assay to accurately call indels, as well as T790M and C797S. So I hope today that you've taken away from this talk a few different things. First, blood-based approaches have become an integral part of the biomarker strategies in drug discovery and development. We've shown that both ddPCR and NGS-based tests can offer the ability to conduct very highly sensitive analysis of the circulating nucleic acid that is extracted from plasma when using liquid biopsy. The Biodesix GeneStrat 52 NGS assay, we've demonstrated feasibility in the Biodesix laboratory for SNVs, indels, as well as fusion and copy number variations. And then we specifically focused on validating those driver mutations in EGFR, BRAF, and KRAF, looking at analytic sensitivity, specificity, precision, and accuracy studies. And we found that there was really nice correlation both between ddPCR and the GeneStrat 52 panel, with a positive predictive agreement of 91.7% and a negative predictive agreement of 100% between the two assays. We are planning to do additional stability studies to continue to define the pre-analytic conditions for the STREC DNA BCT that has been used in this assay, and how that impacts and works with the NGS GeneStrat 52 test. So with that, I would love to thank the Biodesix team, the lead scientist that worked on this project and really generated much of the data that you saw today, Jordan Reese, as well as other individuals in the laboratory for their support in this project. This is our team. This is post-COVID, as you might have guessed from the image. But these are the warriors that are in the lab today, both continuing to deliver tests to inform lung disease and lung cancer, as well as now also running the assays for the SARS-CoV-2. And then finally, also, I'd be remiss not to thank our Thermo Fisher scientific team, both technically just very, very knowledgeable and also very willing to help and support us. So really appreciate all of the work that they've done to bring this test up to the point where it could be implemented in the clinical laboratory and helping to support us through all this work. With that, Natalie, I am going to close the talk and I'm happy to take questions.
Thank you, Hestia. That was a really informative presentation, and I hope the audience enjoyed it as much as I did. I'm just looking in the question and answer box here, and we have a couple questions that have come in. So I'll just get started right away. First question, is the test ready for use in the CLIA laboratory?
Thanks, Natalie. Yeah, good question. It is. So as I walked through the preparations for the validation, you could see that we have all of our SOPs and our training and competency completed for individuals in our laboratory. We have our instruments under control, and we've got our validation assays completed. So we are ready to implement this assay into the clinical laboratory for use in clinical trials, for example, would be really great.
Awesome. Thank you, Hestia. Another question here that we have is, what's the turnaround time of this assay if it were used clinically?
Yeah, that's another really great one. So the end-to-end workflow from when a specimen is received in the laboratory to be able to get a result is about 4 days. So 4-day turnaround time. And then usually we ship overnight. So if we can get the specimen overnight to the laboratory, then from the time the specimen leaves the office to the time a result is available is about 5 days.
Great. Looks like we have another one here. So what type of plasma is the test compatible with?
Yeah, so we have implemented the Streck cell-free DNA BCT for this test. And we really like using those types of tubes that can stabilize nucleic acid for shipment at ambient temperature. And we've already been using that tube with our digital PCR test for quite some time. There's lots of benefits to doing that when you're trying to do prospective studies, for example, or in a commercial environment where you're, of course, a centralized lab servicing physicians all across the US. However, there are instances where pharma actually has banked samples and they may have been collected in other ways, the most common being in an EDTA tube. And so in those situations, we also are able to process specimens from plasma that was collected into EDTA and then separated and stored at -80.
Awesome. It looks like we have one more question here. So just as a reminder to the audience, if you do have any more questions, please submit them now. The question is, I noticed the validation was done on the DNA variants. Does the test have capabilities to run RNA?
Right. So that is perfectly true that the validation of the assay has focused on EGFR, KRAS, and BRAF variants, which are both single nucleotide and small deletion. However, the assay does have capability, and we showed a little bit in the feasibility, to do fusions and to do copy number variation as well. And so I'm eager to also have the opportunity, the samples, which is always one of the biggest struggles, to start the validation process on those variants as they're needed as well.
Thank you, Hestia. So it doesn't look like we have any more questions, but if anyone has any, please put them in the question box now, and we will be able to get to that question. If not, doesn't look like any more are coming in. So with that, I'd like to thank Dr. Mellert for her amazing presentation, and thank you to all of the attendees for tuning in. We will send out the webinar recording to all who've attended, as well as all registrants. Thank you so much.
Thank you, Natalie. Thanks, everyone.